Okay, kia ora everyone. My name is Bree, and this summer I've had the privilege of working on some data, working on Phytophthora pluvialis, specifically looking at its gene regulation when confronted with resistance in Pinus radiata, and particularly looking at it from a computational point of view. So a lot of you have probably had a lot to do with Phytophthora pluvialis, but if you're unfamiliar with it, it is a necrotrophic oomycete that sends out effectors which essentially infect the Pinus radiata and suppress its immune system. So studying this will be beneficial to help us understand some control mechanisms that we can target to treat this disease. Cool. All right, I'm going next slide. Cool. So just a little bit of a background about the experimental design for this study. So I got given some metadata, and this is what we sort of deduced was the study design from it. So there were two pine phenotypes. There was the infection-resistant pine and the sensitive to pluvialis pine. It was taken at 4 different time points. So the data, just the transcriptomic data was collected at these 4 different time points. There were 2 different treatments. So there was the control treatment and then the pluvialis treatment. And then there were 3 different replicates for each condition. So the data that I was given was RNA-seq data, and it was given, and these were the given conditions, the experimental conditions that the data was collected under. Okay, so what did I do? So this is a little bit of a sort of pipeline of what I sort of carried out. So started out with these raw RNA-seq reads from Scion, who kindly provided the data. And then there was 2 sort of pipelines, 2 different kind of approaches that I took. So one approach was using an already built RxLR effector database, and then running this on, with BLAST and blasting the raw RNA-seq reads against this to identify the effectors that were present in the RNA-seq data. Did a bit of filtering, exported it to R and built a bit of a counts table, and then use the metadata and to carry out a differential expression analysis of these effectors. The other pipeline I took which is the top boxes. Sorry, I don't think my my mouse can't point to the things, otherwise it would be a little bit easier for you all to see. But so that was sort of the bottom pipeline. So the top pipeline, I did a bit of quality checking, and then I assembled a transcriptome with Trinity, and then clustered with MMSeqs and mapped these trim reads using a transcriptome tool called Salmon, and then also exported to R for differential expression analysis. So just a little bit about sort of the top pipeline. So the reason that I assembled this transcriptome and used transcriptomic data as opposed to genomic data is because, of course, with transcriptomic data, you have spliced out the introns, so you're only getting the expressed portions of the DNA. However, if you were using genomic data, of course, you have these exons, these introns in as well, so you're not necessarily getting the data, the effector data of what has actually been expressed. And also wanted to build this transcriptome rather than just get one off the internet and just use that because I wanted to look at these specific experimental conditions that they use to gather the data and build a transcriptome from that. Otherwise, it could be slightly different. But I did end up by combining this one with a reference transcriptome to build sort of a larger database that I could then carry out my differential expression analysis with. So anyway, in terms of a few results, tables and things, managed to build an effector counts table with the effector protein database to get kind of numbers of effectors per transcript. Then was able to carry out differential expression analysis on the different clones to kind of get whether they were significantly upregulated or downregulated. And that's what you can see in this graph here. So anything above the log 2-fold change of 0 would be considered upregulated in the susceptible pine, and all the green dots are the ones that are significantly upregulated compared to the resistant pine. And as you can see, there's not a lot of downregulated genes in the susceptible pine, and so this probably would indicate that in susceptible pine, these effectors secreted by the pluvialis are not suppressed. So pluvialis is able to produce more effectors, whereas in the resistant pine, it's likely that the pine is suppressing the expression of these pluvialis effectors. So essentially stopping this infection and suppressing the immune response. So yeah, that's a bit of an overview about sort of about my project. I'd like to give a big thank you to everyone involved. Thank you to Bioprotection New Zealand for your Aotearoa, for your support during this project and for funding this project. I'd also like to say a big thank you to Craig and Claudia for supervising this project, and a big thank you to Cyan for the data. And does anyone have any questions?
Awesome. Thanks, Bree. Great talk. So has anyone got any questions for Bree?
Bree, just can you hear me? Obviously, being resistant to Phytophthora pluvialis seems to be a real advantage. Why aren't all pines resistant? Why haven't they all evolved this ability to resist the widespread pathogen? Why are only some clones resistant and others aren't?
That's a really good question. I think it will come down to whether or not, I guess, they have the ability to suppress a certain the expression maybe of these effectors, or if they've got sort of beneficial adaptations to kind of combating these effectors. And yeah, I'm not entirely sure, but I guess that's something that could be sort of future kind of looked into in this study, and like studying kind of how and why these effectors are suppressed in certain pine are able to do that, and whether or not they're that can be like, I guess, altered or changed. But yeah, no, that's a really good question.
Okay, thanks.
Any other questions for Bree? I'm just searching through the people there to see if we've got any raised hands. I might ask you one, Bree. So what proportion of your effector catalogue was actually expressed in the susceptible pine line? Was it a good portion of them, most of them, all of them, or was it just a select number?
So you're saying like, of all the effectors expressed,
All the effectors from your catalogue that you had, your sort of from your starting point that you had looked to map reads to, were a good portion of those expressed? Was it what sort of proportion of those from your original database, I guess you could say, were expressed?
I'm not sure I kind of understand your question. So you're saying like, so I had this database, and what proportion were actually expressed in the
Susceptible.
in the susceptible?
Yeah, so did you just get like a few effectors? I don't know what your total number was that was expressed.
Yeah.
You
I
might not
can't
know, which is
off
fine.
the top of my head. Sorry, I can't tell you off the top of my head. But there were a lot of effectors expressed, but it was a large effector database, so I doubt it would have been. Yeah, there were a lot expressed, but the database was also huge. So
Okay, no, that's all good.
and I'll tell you off the top of my head what the proportion would have been.
No, that's all good. Rebecca?
Yeah, hi Bree. Can you see
Hi.
me? So I was wondering if you noticed any difference in the time points and the effector expression.
Yeah, so this is something I'm actually looking at the moment. I was carrying out differential expression analysis on the different time points. Actually, obviously at time point 0, there was actually nothing expressed. But
Good.
that's kind of what you'd expect. But with everything else, I was trying to run it on differential expression on the separate time points. But I kept running into errors in my code because it told me there wasn't enough data. I'm not sure why. Still kind of troubleshooting that at the moment. Enough data for each of the individual time points.
Yeah.
So that's kind of the next steps, is some of the next steps as well, is we'll be looking at those time points, sort of troubleshooting why it's not letting my code run.
Yeah, I can
because-
probably help you with some of that. We can chat about it later.
Okay, cool. Yeah, no, sweet. That'd be great. Thank you. Yeah.
Okay.
All
Great
right.
talk.
Thanks, Rebecca. Thanks, Bree. That was a great talk.